It's my distinct pleasure today to introduce our speaker, Dr. Kory Lavine. Dr. Lavine, let's see how to define him. I would say he is the epitome of today's modern physician scientist. A modern physician scientist who really takes the most advanced molecular techniques and applies to understand the fundamental basis of cardiovascular diseases. Really, in this regard, I would say Kory is an epitome. And we're very proud to have him here today as our speaker. I'd say, yeah, Kory, from the very beginning of his life, was one of the most fortunate people that knew what he wants to do in life. When he joined the medical school, he started the MSTP program at WashU. And I tell you, even during that time as a medical student, it is a stellar job. He did join Ornitz's laboratory and together worked on FGF and delineated the role of fibroblast growth factor in coronary vascular development and myocardial regeneration that he successfully published in Developmental Cell, I believe, let me see, quick cheat 2005, that's correct. And then he followed that by physician scientist training program also at WashU, became an internal medicine doctor and became a cardiologist focused on heart failure, became a heart failure specialist and developed expertise in heart transplantation. So heart transplant physician and heart failure physician at WashU. And meanwhile, he built his career, a research career, by doing a postdoc with Doug Mann, our great friend Doug Mann. He during that time he did notable contribution to our understanding of a distinct set of macrophages of different origin, different biological function that are involved in both pediatric and adult heart failure. And I believe he published that in PNAS. So he had, from the very outset, he had an outstanding, stellar, really poster career, I would say. Is a brilliant program that he set up and succeeded. And then he was appointed as faculty in 2014, and soon he rose in less than 10 years, rose the rank of academia to full professor. He's currently holds the position of Alan A. and Edith L. Wolf Professor of Cardiology. He's also Professor of Developmental Biology and Immunology and Pathology at WashU. He is the Director for Center Cardiovascular Research at Cardiovascular Precision Research Initiative, and also co-development of developmental regenerative and stem cell biology at the graduate school at WashU. As I indicated, really, his research program from the very outset has been stellar and exemplary. He has published over 200 highly impactful papers. He has in major biomedical journals. He has been one of the Clarivate highly cited scientists since 2022. He has, despite his relatively short career, he has an H-index of 57 and over 50,000 citations to over 200 publications that he has published in the major biomedical journals. His research program, in addition to delineating the role of immune cells in heart failure and cardiovascular pathology, he is also focused on developing imaging tools and techniques to image this inflammatory cells in the heart. It's a program that he has been successfully running. In addition, he has a branch of his research program that focuses on developing small molecules and antibody-based therapeutics for heart failure. His research, as would you expect, has been funded successfully by R01 grants from NIH. He is also the recipient of Outstanding Investigator Award from NHLBI, and he is a member of the Leduc Foundation, which is a wonderful program that Leduc has been doing over the past 20-some years. Is a great opportunity to really do transatlantic collaborative research program that Dr. Lavine is currently conducting. He holds 4 patents for his scientific discoveries. Dr. Lavine has been recognized for his scientific accomplishments by numerous awards. I would not mention the list, but I have listed in his biography. One of my favorite is Burton Sobel Award. Dr. Sobel was a pioneer clinical investigator at WashU. Some of you know him. He is no longer with us. He was the pioneer in the use of TPA for acute myocardial infarction. And in addition to Sobel Award, he has several other awards, including Career Award for medical scientists from Burroughs Wellcome Foundation, which is a fantastic program that Burroughs run, and I'm very proud of them for that. He was inducted to ASCII Society in 2017, or sorry, 21. And also, impressively, he is the recipient of the Presidential Early Career Award for Scientists and Engineers in 2019. He has several other awards that I've listed in his biography. We are most delighted and honored to have Dr. Kory Lavine as our speaker at the IACS webinar series. Our panelists are Michael Schubert. Michael Schubert is the past president of IACS. He is a professor of physiology and pathophysiology. He has endowed chair in fibrosis research at University of Manitoba, Winnipeg. Our next panelist is our famous colleague, Nick Frangogiannis, also one of the highly cited scientists. He's professor at Department of Medicine, Department of Microbiology and Immunology, also holds an endowed position in cardiovascular medicine at Albert Einstein College of Medicine. Our next panelist is Dr. Priyantash Gurha, who is a Welsh and Dunn Foundation scholar. He is assistant to associate professor at our center at the UT University of Texas Health Science Center at Houston, and a brilliant young scientist. Finally, Timothy McKinsey, one of the pioneers in also myocardial fibrosis, who is professor in the Department of Medicine and Division of Cardiology at UC Denver, an endowed position in cardiology, Joseph Rose LaConte Cardiology Endowed Chair. He's Associate Division Head for Translational Research and Director of the University Consortium of Fibrosis. With that, as I indicated, Dr. D.K. Davendra Agrawal is our co-host. He will say a few words about the program at the end of the webinar. And then our program director, I introduced Professor Natalie Hernandez. We're grateful to her. With that, Dr. Lavine, we are most delighted to host you and grateful to you for participating in our webinar series. Now the podium is yours, Dr. Lavine.
Well, thank you so much for the invitation to present and the very kind introduction. I think I can retire now. There's nothing more to do based on that description, but that's far from the truth. You mentioned Dr. Sobel briefly. I'm actually in Bert's prior lab space, which is really special, and that's been an honor to even be able to walk in any of his footsteps. So what I like to do today is talk a little bit about some newer work from our group focused on cardiac fibrosis and some new insights into potential targets and therapeutic approaches that have come from these endeavors. All right, so here, my disclosure is nothing that's particularly relevant for today's talk. And then this is the outline of the topics that I hope to hit on. I'm going to first begin by introducing some concepts around cardiac fibrosis and fibroblast origins. then talk about our work from using multi-omics single-cell technologies to profile human hearts to identify what fibroblasts exist within the myocardium. Speak a little bit about the relevance of mouse and cell culture models to study these interesting populations, and then spend a bit of time sharing work on targeting one population of activated fibroblasts that's marked by fibroblast activated protein, or FAP, and discuss how these cells come about with crosstalk from the immune system and transcriptional events that govern their specification and maturation, and then end with our approach to clinical translation of those findings and how to try to bring some of this work forward to the clinical setting. So as an introduction, I think we all know that fibrosis is an important part of cardiovascular disease and heart failure. And we all recognize it's a major issue in patients with ischemic cardiomyopathy. So compared to healthy controls shown here, individuals with ischemic cardiomyopathy have replacement of myocardium. The cardiomyocytes are in kind of dark pink here, with fibrotic material that's in the light pink color. But I think what's a little less appreciated is that fibrosis doesn't just occur in ischemic disease, but also occurs in non-ischemic forms of heart failure. And this is a typical specimen obtained at the time of transplant. A patient with non-ischemic cardiomyopathy, even individuals with sarcomere mutations, have quite a bit of fibrosis within their myocardium. So it's prevalent across the spectrum of heart failure. And not only is it present, but it's associated with poor prognosis and associated with lower rates of survival. So many people use cardiac MRI and like gadolinium enhancement as a surrogate for fibrosis. And in many different studies, this is just two as an example, having evidence of fibrosis by late gadolinium enhancement portends a poor survival. And the extent of fibrosis, whether it's mild or severe, is also associated with poor outcomes. So it's there, and it's a bad thing to have in your heart, and we really have very few therapies that target cardiac fibrosis. And so a lot of attention has been spent on understanding how fibrosis occurs in the heart. And much of what we know is derived from animal models. And so this is a picture made by Michelle Talquist and Jeff Mulkenton that summarized the field in 2017. It's not just their work, but work of the collective field, including Eric Small, Tim McKenzie, and many others. And the thought was that, cardiac fibroblasts exist within the myocardium. They're these resident fibroblasts. Many of them come from the embryonic epicardium, and they differentiate during development to form the interstitium of the myocardium. And then there's some fibroblasts that come from the embryonic endocardium and adult endocardium. They undergo an endothelial to mesenchymal transition and become fibroblasts. These fibroblasts get activated during times of injury, stress, and disease to give rise to an activated population that over time has been lumped into one term called myofibroblasts. But as I'll show you shortly, likely represents a compilation of different states and subsets. Over the past years, there's been other sources considered for activated fibroblasts or myofibroblasts. These include pericytes, endothelium, and smooth muscle cells, as well as circulating cells such as monocytes, bone marrow progenitors, or circulating fibrocytes. Their actual contribution is thought to be minimal in mouse models of pressure overload and ischemic injury, but we really don't know as much in rare forms of heart failure or in humans. And we don't actually know if any of these things are true within the human heart. And that was really the driver for our involvement in this field, was to try to understand what the fibroblast populations are that exist within the human heart, how they get activated, and which subsets contribute to disease. And so this project began with a reverse translational approach where we profiled human tissue, either donor hearts, so these are hearts that are undiseased, but and came from individuals that were brain dead, whose organs were not used for transplant. Patients who had an acute myocardial infarction, chronic ischemic or chronic non-ischemic cardiomyopathy, they're undergoing LVAD or heart transplantation. So we had an opportunity to obtain these tissues, and we performed site-seq, which is a combination of single-cell RNA sequencing as well as profiling of the surface epitome, about 279 proteins that exist on the cell surface. And then we also performed multi-omic sequencing, which is a combination of RNA-seq and ATAC-seq to give an idea of what RNAs are expressed in each cell and where the chromatin might be open. And we use this as an atlas to try to understand what the populations are within the myocardium. So this is a typical representation of one of those experiments. This is specifically from the site-seq data set, which does not include cardiomyocytes, which are difficult to digest from the human or mammalian heart. And instead, it contains many of the stromal elements that exist within the myocardium. And you can see we can identify common populations like endothelial cells and fibroblasts, and then really rare populations such as mast cells, B cells, or even glia that are present within the tissue. And if you look at how these subsets change across the different disease conditions we looked at, as one would expect, immune cells like myeloid cells and T and NK cells expand in acute MI compared to donor hearts. That's a nice control to see. And we're focused on fibroblasts for this presentation, and you can see that the number of fibroblasts compared to donors increased in acute myocardial infarction, ischemic, and non-ischemic cardiomyopathies, as you would expect from the pathology. And so that was expected. If you ask where the gene expression differences lie when you compare donors versus these different forms of heart failure, this is ranked by the total number of differentially expressed genes. And you can see that fibroblasts were by far the most, or the number one population where gene expression differences were evident or observed. And so we decided to dive into this in much more detail to understand how these populations shift at the single cell level. And so that's what this data looks like. This is a typical UMAP plot of cardiac fibroblasts. Each color represents a different transcriptional state of cardiac fibroblasts. You could spend an hour talking about whether those are different cell types, different cell subsets, but we're really just looking at transcription. So we call them different transcriptional states of fibroblasts. And I'm not going to talk about all of them, but I'm really going to focus on two of them today. One is on the top in this tan color, which is population F9, which is marked by fibroblast activated protein and periostin. And the other one is in dark blue, which is the population many of us are very familiar with, which is the myofibroblasts. And so when you look across disease conditions compared to donors, we can see that these FAP or periostin positive fibroblasts really expand from almost not being present in donor hearts to being a reasonably prevalent population in acute MI and remain elevated in chronic disease such as chronic ischemic and non-ischemic cardiomyopathies. And you see that the number of these cells is actually slightly more than that of myofibroblasts, which also expand in acute myocardial infarction, ischemic and non-ischemic cardiomyopathies, shown in the dark blue here. The FAP positive and periostin positive fibroblasts are indeed a distinct population. Here's their FAP expression at the RNA level in red and the protein level in yellow. You can see they're pretty specific to this tan population and really not found in the myofibroblasts shown here, which express things like smooth muscle actin and other canonical markers. And then this FAP positive population also expressed periostin and then markers of activated fibroblasts such as Ty1 at both the RNA and the protein level. So this isn't a sequencing artifact, but you can clearly identify these cells in tissue using the FAP antibodies shown here. You can identify these activated fibroblasts in acute myocardial infarction, and this is a different set of spatial transcriptomic data that we obtained from Raphael Kramen in Aachen, Germany, as an orthogonal data set, which showed us that they're prevalent early after infarction, shown at 2 days and 11 days after MI, and then were lower but remained elevated for years after infarct compared to donor controls. And this work was nicely complemented by John Epstein and Ken Margulies, showing that FAP positive fibroblasts were also upregulated in chronic forms of heart failure, as well as cardiac sarcoid, giant cell myocarditis, and other inflammatory cardiomyopathies. And it's wonderful to see many different groups kind of rallying around particular markers. So one of the key questions that we want to ask was, where do these cells come from? And are they distinct from myofibroblasts? And that's an important distinction. because we understand that myofibroblasts are important for not only scar and poor outcomes, but also the structural integrity of the heart. So we want to understand that relationship. So one way to do this is to take advantage of the single cell sequencing data and use modern informatic technologies to try to create predictions or inferences of how these different clusters are connected to each other and where their cells come from. We use two orthogonal approaches when we do this. We use an algorithm called Palantir, which uses a Markov chain model and a probabilistic algorithm to predict how one cell may give rise to other cells. And then we use a second completely different approach called RNA velocity, which allows you to connect different clusters by looking at pre-mRNAs. And the idea is if cluster B comes from cluster A, then cluster A will start expressing pre-mRNAs that are markers for cluster B, which makes, I think, intuitive sense to most biologists. And based on the number of pre-mRNAs made, you can predict how fast it's moving to that area. So what I'm showing you here is a UMAP or force-directed layout representation, much like a UMAP plot, looking of our Palantir data. And the Palantir approach identified or predicted two different end states. One's called F2, which is our myofibroblasts, and a separate end state called F9, which is the periostin positive fibroblasts, which are shown over here. And then the starting state were the naive fibroblasts or ground state that started in this area of the plot. And then we can overlay the vector field from the RNA velocity data. And that vector field shows that if you start with a red population, you can move towards F9 at the bottom of the screen, or you can move towards F2 at the top part of the screen, and the vectors circle around the terminally differentiated states. And that's a nice way to show with two orthogonal approaches, a shared prediction that the myofibroblasts and periostin FAP positive fibroblasts are likely mature fibroblast populations that are distinct. So how can you prove that? So the first thing we did was to try to stain for markers of each of these populations to see if there's any overlap in human hearts. And we found surprisingly very little. So in human hearts, periostin was pretty exclusively expressed in fibroblasts. And we could find areas where fibroblasts expressed smooth muscle actin. And in these, as you can see in these insets, there were very few cells that co-express both green smooth muscle actin or white FAP. We could take mouse spatial transcriptomic data and ask where these cells were located. And we found they were largely located in different places. So the myofibroblasts were located within the core of the infarct, and the periostin FAP positive fibroblasts were located more in the border zone. So they may be spatially distinct as well. And to really try to prove this, we made an FAP ER-Cree mouse so we can trace the fates of activated FAP positive fibroblasts and ask what they can become. And what we did here is we took these FAP ER-Cree mouse, crossed a reporter, did Ang2-PE infusion as a model of hypertensive heart disease. This also works if you do MI. Give tamoxifen after injury, and look 2 to 4 weeks later and ask what these FAP positive cells might become. And we stain for both TD tomato, periostin, smooth muscle actin. And what we learned in this composite image is that almost all of the red cells were also periostin positive, as we would expect from the human data. And there was almost no overlap between FAP and smooth muscle actin, and that's quantified right here. And so this tells us in a lineage tracing approach that FAP positive fibroblasts did not become a myofibroblast, that they remained a distinct population over time, which adds to the argument that it represents a different lineage. So if we want to understand what these cells are and what they do, we need a model to look at them. And so there's, I think, a big debate in the literature, which has been amplified by US politics and the NIH, of whether we should be using mice as models, or we should always be looking at human cells in a dish. And we thought this would be a nice opportunity to leverage single cell sequencing data and ask what is a good model of this population and other fibroblasts located within the myocardium. And so what we did is we were able to use our own and others single cell sequencing data sets from mouse models of cardiac injury, all the ones that we all use, LAD occlusion, transaortic constriction, Ang2P infusion, grab the fibroblasts, and then we map them onto our human data. And when you do that, you can identify mapping scores to give you an idea of how well the cell's mapped and how confidently you are with not just looking at one or two genes, but looking at thousands of genes, how similar these populations might be to each other. And so this is an example of one of those analyses where we took LED ligation data. This is from Nadia Rosenthal's group, and we mapped it onto the human data set. And you can see many of those populations are present, including our FAP periostin population showed up here. There is one population that's not present in mouse hearts, and that represents adventitial fibroblasts that live around coronary arteries that we see in humans. And that may represent an important distinction between mice and humans. But the nice thing is the FAP positive cluster was there. It had a really good mapping score, close to 1. And this population increased after MI, shown in tan here, and went down a little bit, but remained elevated 4 weeks after infarction, much like we saw in humans. And the same was true of myofibroblasts, which is shown in the dark blue. This mapping nicely worked for Ang2PE and TAC models. And so it gave us the interesting argument that mice may be a good model for human cardiac fibroblasts. So there could be use of mice. In contrast, when we looked at human cells in a dish, whether it's either using primary human cardiac fibroblasts, primary human dermal fibroblasts, or immortalized human cardiac fibroblasts, we actually saw very poor mapping of the FAP positive population to these cell types, whether they're cultured by themselves with IL-1 beta, or even, and maybe a little bit better with TGF beta, but the mapping scores really weren't great, which tells us that human cardiac fibroblasts, when they're cultured in plastic, with the growth factors that we're using, we're not really recapitulating the same types of activated fibroblasts, but those studies are really good at recapitulating myofibroblasts. So they're not totally unuseful. They're just not useful for this particular population. And I'm not making the argument that the cells can't become FAP positive fibroblasts. We just don't know how to do that yet. Whether they need to be cultured in a particular stiffness, need to have other cells around them, or a particular extracellular matrix remains open questions. This is what, for the computationalist listening, this is what the mapping scores look like. For the mouse models, we saw excellent mapping. Mapping score of 1 is perfect mapping. And for all the cell culture models, the mapping was quite poor, all below 0.5. Okay, so now that we have a model and a mouse where we can mark FAP positive fibroblasts with confidence, we want to know whether they're really causative or in the causative pathway for disease. And so we did a number of different experiments. I'm going to show you an ischemia perfusion experiment where we've taken control mice that are FAP ER-crew with a reporter, but also express a diphtheria toxin receptor, so we can give diphtheria toxin and deplete them. And so when we do this after MI, we can see a good number of FAP positive fibroblasts that locate within the infarct and border zone. And when we deplete them, we can get about a 75% depletion with this model. And when we do that, we see a really nice improvement in cardiac function. So this is a control animal, echoed 4 weeks after ischemia or perfusion injury. You can see the nice infarct, LV dilation, reduced function. And when we deplete the FAP positive fibroblasts, the infarct got a little bit smaller, the function got quite a bit better, and the LV volumes got better. And there was no rupture and no death within this model, and no rupture or death within a chronic ligation model either, which tells us that this population is not important for the structural integrity of the heart when we deplete it, but is important for remodeling. And this is the quantitative data. You can see that there's really a robust improvement in ejection fraction and reduction in LV diastolic volume and reduction in the infarct area when normalized to the total area at risk, or if you don't normalize, you also see a reduction. Okay, so what are the therapeutic opportunities here? And so it's clearly possible that removing the cell type could be of benefit. And so there's lots of options to do this. There's ways to deplete cells in vivo. I'll just mention this in passing today, but we talk about this in a different time. The two areas I really want to focus on are there opportunities to interrupt cell communication that may be important for specifying this population? And are there opportunities to prevent the differentiation of these activated fibroblasts, and really talk about the latter two. And these may be nice therapeutic options. All right, so I'm going to start with the cell communication. And so to understand how these cells get activated and who's communicating with them, you need to know who their neighbors are. Who do they live with within tissue? And so we took an unbiased approach to this. We took the atlas that we developed. This is now the single nucleus RNA sequencing atlas, so it includes cardiomyocytes, which are shown in red here. We have really every population you see within the heart. We did spatial transcriptomics, so we could map these cells into space and create neighborhoods and use statistical approaches. This is an example of a Pearson correlation network where you can identify niches of cells that are commonly located together. So we're all used to the vascular niche where there's endothelial cells, but there's also cells and pericytes. You can see that nicely here. And then we also were able to identify a niche that included the FEP positive fibroblasts and a lot of myeloid cells, which I'll show you are macrophages in a second. When you look at these niches as a unit, niche 0 is what we're going to focus on, which is called, we named the immunofap niche. This niche was enriched in collagens, decorin, and a number of proteins found in, an RNA, sorry, found in activated macrophages, as well as activated fibroblasts. And if you look at the pathways in this niche, interestingly, you didn't see a lot of TGF beta, but instead we saw NF-kappa B and TNF, JAK and STAT signaling and some VEGF, which are largely inflammatory pathways. All right, so what cell types in this niche could signal to each other? And so to prioritize the possibilities, we used a algorithm called NicheNet, which can make predictions about cell communication. And so the way this network or this approach works is it takes in account the ligands expressed on cells, the receptors expressed on your target cell, but also the gene regulatory networks and the target genes that are downstream of the receptors. So it gives you an idea of who could be signaling, who could be receiving, and who's actually receiving signal. And so we employed this framework using fibroblasts as the receiver. We predicted, pathway predicted, that TGF beta and IL-1 beta were the two most important ligands to fibroblasts. TGF beta was actually an autocrine pathway going from fibroblasts to myofibroblasts, and IL-1 beta was a paracrine pathway predicted to go from macrophages to fibroblasts, and that's the one that I'm going to focus on. There's many more that actually came out of this that I think are important areas to study. And so I don't want to leave you thinking that IL-1 beta is the only pathway that's important, but the one that we decided to focus on initially. So where's IL-1 beta expressed? IL-1 beta is exclusively expressed in myeloid cells or macrophages. And when you subcluster these macrophages, IL-1 beta is expressed in a collection of subsets that are marked by the expression of CC chemokine receptor 2 or CCR2. And so that's shown in UMAP space when actually looking at the individual cell types. For people used to looking at this data, these are monocytes here. These are intermediate monocytes and early differentiating macrophages. And then these are MHC class 2 high macrophages shown up here. If you look at where these transcripts co-localize in space and spatial transcriptomics, they're both localized at the edge of the infarct zone, both IL-1 beta and CCR2. We can then do immunostaining to validate this. And so this is immunostaining of specimens for CCR2, FAP, and a marker of macrophages CD68. You can see that they kind of lump together in these neighborhoods. And then you can look at this in a more unbiased way of asking whether cells co-expressed periostin and IL-1 and whether there's periostin positive fibroblasts co-localizing with CCR2 positive macrophages that make IL-1 beta. And we saw a nice distribution that fit a line. And then we could identify similar collections of cells in mouse models. And so this tells us that these cell types are co-localized, and there's a potential ligand IL-1 beta that's enriched in these populations. One of the, I think, main disadvantages of looking at these human tissues is this represents end-stage disease. And so we want to make sure that these interactions are also present in earlier forms of heart failure. And so one approach that we've used to investigate patients when you can't take out their heart or can't biopsy them is using molecular imaging to identify these populations and where they may be present. And so these are two imaging tracers that we have developed. One's against CCR2. It's a gallium-based PET probe. And another one is fibroblast activated protein, which is also a gallium-based PET probe. Gallium is really nice because it has a very short half-life, and so we can image patients on successive days. And so this is an example of an individual with ischemic cardiomyopathy that was recently in the hospital with heart failure who has frequent readmissions. And you can see that there's nice co-localization of CCR2 shown here, and then FAP shown here. And we've seen this across lots of different patients. And so it gives us increased confidence that these cell types are located in similar diseased areas of the human myocardium when it's not just end-stage disease. All right, so how can we prove that IL-1 beta signaling to fibroblasts is important for FAP-positive fibroblast fibrosis and cardiac dysfunction? And so we tackle this by knocking out IL-1 beta in the CCR2 positive macrophages. We developed a flow cytometry assay for FAP, so we could do this in a bit more unbiased manner. And we employed an Ang2 phenylephrine hypertensive heart injury model for ease of use. And when we do this, we can see a nice accumulation of these FAP positive cells that reduced by more than half when we knocked out IL-1 beta in the CCR2 positive macrophages. This led to improvements in cardiac function, and we focus just on ejection fraction and fractional shortening here. Compared to controls, when we did the Ang2P model, we saw a reduction in fractional shortening, or EF, that normalized back to baseline when we knocked out IL-1 beta in the CCR2 positive macrophages. And this is an example of some of the M-mode images. And then we were able to reduce fibrosis as well. We can see an identical phenotype if we knock out the receptor, IL-1 receptor, in fibroblasts. In this case, is using a derma-1 Cre, which is a nice marker of epicardium and epicardial-derived fibroblasts. And in this context, we also saw a reduction of FAP-positive fibroblasts, marked reduction in fibrosis, and we even saw improvements in cardiomyocyte hypertrophy to some degree and improvement in function. And then we can recapitulate these findings using the IL-1 beta neutralizing antibody. And so here's a tool molecule that's similar to canakinumab, where we gave it to mice that received the Ang2P pumps. With this antibody, we can see a reduction in the FAP-positive fibroblasts, as you saw in the knockout animals. We can use our PET tracer, and we saw a reduction in the FAP PET tracer as well, which is a nice orthogonal approach, and a reduction in fibrosis. And so these data are great in telling us that IL-1 beta from macrophages of fibroblasts is important in their differentiation. But we want to go a little bit deeper and understand how that might occur. And is it more than just differentiation? Is it actually their maturation as well? And so we did same experiment. We gave IL-1 beta neutralizing antibodies, and we looked at day 7, and we divided fibroblasts based on flow cytometry into a ground state or a state that is somewhat activated. And then we did sequencing on each of these libraries, a ground state and activated state, and divided it into animals that received the isotype control or anti-IL-1 beta. And so I'm going to focus you over here. And so when we focus on the activated fibroblasts, when we have the isotype group, we have high levels of FAP and collagens. And when we gave the anti-IL-1 beta antibody, those reduced. And if we look at typical markers of activated myofibroblasts, things like transgelin, smooth muscle actin, SFAP, these were actually increased with the anti-IL-1 beta antibody, suggesting that we're able to toggle the fate from an activated FAP-positive fibroblast to a myofibroblast, which we thought was actually quite interesting. We also identified some interesting effects with type 1 interferon signaling that we don't understand yet, that I just wanted to share. And then we could map this onto our human data and show that the fates that the IL-1 beta neutralizing antibody was driving the fibroblast towards were relevant to human. And so in the isotype control, they're largely associated with the periostin FAP fate. And when we gave the anti-IL-1 beta antibody, they were more similar to the myofibroblast fate. And this is what that data looks like and that data quantify. So collectively, this information tells us that CCR2 positive monocytes and macrophages make IL-1 beta and probably many other factors that signal to fibroblasts and promote the specification and maturation of the FAP and periostin fibroblast population. I think this is a really cool finding because it's immediately clinically actionable. We have great drugs already, available for patients. They're anti-IL-1 beta antibodies like canakinumab. There's an anti-IL-1 receptor antibody that's already been through clinical trials and is safe in humans that was used in osteoarthritis. There's IL-1 traps we all use for recurrent pericarditis like rilonacept. And I think there's an opportunity to develop bispecific antibodies to make these therapies safer by targeting the inhibitor to the area or the cell type that we want to inhibit to reduce the risk of infection and potentially increase benefits. And so I'm hopeful that we'll see additional clinical trials in the space and additional therapeutic innovation to make these therapies more effective and safer over time. So I'm going to switch gears for the last few minutes of the talk and discuss what are transcriptional regulators of FAP positive fibroblasts. What could we do to potentially, target their differentiation outside of just IL-1 beta signaling? So to approach this, we did a differential expression analysis that compared myofibroblasts to these periostin FAP positive fibroblasts, took those differentially expressed genes and asked what are the transcription factors that were predicted to regulate those genes. So top ones that came out were MEOX1, that's already been published to promote activated fibroblasts specification of the heart by Deepak Srivastava and Mickey Alexanian nicely. And then we also identified RUNX1 as one of those transcription factors, and that's the one I'm going to talk about. RUNX1 is a really cool gene that seems to co-localize with periostin and MEOX within a gene regulatory network that we identified using site-seq and ATAC sequencing together. This is an orthogonal single-cell data set from a different group of patients, and we can see that FAP, periostin, RUNX1, and MEOX kind of all localize together. And if we do an in silico perturbation to delete RUNX1 and predict how cells would shift in space, we find that if we either focus on RUNX1 or just ask what are the top transcription factors that would move a cell away from the FAP periostin state and towards another state, and here blue means away and red means towards, RUNX1 was in the top 5 transcription factors that was predicted to be important for maintaining or specifying the FAP periostin state. So I thought that was really interesting. And so we moved forward. This created enough activation energy to breed mice together and asked whether RUNX1 was important for fibroblast activation, at least in the marine heart. And so this is that experiment. We took RUNX1 flox mice with a fibroblast ER-pre. We induced deletion 7 days before IR injury, and then we looked 4 weeks later. So this is a control heart. See the nice infarct and reduction in function. And when we delete RUNX1 just in fibroblasts, we can see the function is better, the infarct smaller, and the volumes are better as well. This is the quantified data. Just 1 day after the infarct, things were the same, and 28 days later, the EF is quite a bit better. We saw a much smaller scar based on either Picroceros red or trichrome, quantified here. And we even saw less cardiomyocyte hypertrophy, suggesting that RUNX1 is actually a really good target in fibroblasts for preventing and suppressing remodeling. We could also see this in our Ang2-PE model where there was reduction in fibrosis. So these are the controls. This is when we deleted RUNX1 with the fibroblasts create. And if we look at what these fibroblasts become when we knock out RUNX1, we can see we find a reduction across many different activated fibroblast markers, those that are important in periostin positive cells like thrombospondin, LTBP2, and COMP, but also smooth muscle actin, so it may be affecting myofibroblasts as well. And then this is the single cell sequencing data from that experiment showing population 3 co-expressed RUNX1 and periostin, and this population was reduced by about 30 or 40% at the total cell level and greater when we look at the actual gene expression within it when we deleted RUNX1. And so let me tell you a little bit about how RUNX1 works, because most people would hear about a transcription factor and they'd say there's no way that you could drug a transcription factor, right? That's interesting, but what are you going to do? So, but RUNX1 is an interesting one where there are opportunities. So this is how RUNX1 normally works. So RUNX1 is shown here. It has a few cofactors. One's core binding factor B, which this interaction is really important for both stabilizing RUNX1 and binding DNA. And that plays a role in both its repression and activation functions. RUNX1 also has a number of co-activators or co-repressors. One of its key co-activators is ETS1, and that binds in a completely different interface down here that is not shared with the co-repressors. And that's encoded in exon 6. We think represents two important interfaces where RUNX1 could be inhibited, and that could be used as a therapeutic. And we're really excited about doing that. We're interested whether we could target these areas to reduce risks, effects on hematopoiesis, and improve benefits. And so this is some of our sequencing data that implicates ETS1. And so if we look at all of the RUNX1 regulated genes in our mouse model and we delete RUNX1, we did multiome RNA and ATAC-seq, we get nice peaks. And if we ask what are the genes regulated by RUNX1 and what are the transcriptional motifs of, in the enhancers and promoters of those genes that are regulated, ETS1 is the number one hit, both ETS1, ETV3, and many others, which really suggests that RUNX1 and ETS1 may be acting as partners. And so we've recently obtained mice that cannot interact, that RUNX1 can't interact with ETS1, and we're profiling that now to understand whether that's the key interaction that's important for cardiac fibrosis, and maybe that's not the interaction that's important for the hematopoietic effects of RUNX1. And so the therapeutic approaches that might exist in targeting RUNX1, one is to consider siRNA therapies. That's something that's been out there, and there's now ways to target siRNAs to particular cell types of interest using sugars or antibodies. And then there's a small molecule approach where we can try to identify small molecule binders that disrupt these key interactions, RUNX1 and core binding factor B, or RUNX1 and ETS1, or simply make it degrader. And so this is an example of how this could work. This was actually done with Tim's lab. And so this is using a tool compound called RO5335, which interrupts the RUNX1 core binding factor B interaction. And so when this is done in culture and fibroblasts are stimulated with TGF-beta, you can see there's nice contraction in the matrix compared to control. And RO5 reduces that quite a bit. So that suggests that it's active. And when we use this compound in vivo, we can see improvements in ejection fraction after an infarct and reductions in fibrosis and hypertrophy. This was done in collaboration with Florian Leuschner's group in Heidelberg. And this is even useful in a cardiac recovery setting. When we do MI, we wait 3 weeks or so, let everything remodel, give the drug, and then echo 12 weeks later. And when we do that, this is a vehicle control. You can see the infarct expands over time. And this is an animal that will receive RO5 and then later receives RO5. You can see that instead of the heart getting bigger, everything gets smaller and function improves. And this is what the quantitative data of that looks like here. You can see that the LV volumes increase in the controls and reduce in the animals that receive the inhibitor. And we see our ejection fraction improves in the RO5 animals and reducing the control. So this works across the entire time course, whether it's in the acute or the chronic setting. So we're particularly excited about this approach. And we've just launched a DNA-encoded chemistry screen, which is a really cool technology that I just want to share for like 1 minute that people may not know as much about. So we always think of drug discovery as being a really difficult thing that's done in pharma. I usually picture stacks of 96 well plates and hundreds of people and a lot of money. But this actually can be done really efficiently today in a test tube. And the way this works is by taking your protein of interest, in our case, it's RUNX1, incubating it with a DNA-encoded library where each compound is barcoded. These libraries contain billions of compounds rather than just a few 100,000, and simply doing a pull-down assay to ask which compounds interact with your protein. And you can determine that by sequencing the barcodes, because each barcode registers a particular compound. And we've done this, And we've identified novel binders that interrupt the RUNX1 cord binding factor B interaction and the RUNX1 ETS1 interaction that we're excited to use to probe the functions in our animal models and hopefully be able to advance a few compounds to the clinical setting. So I'm going to end with one comment about clinical translation. I think translation is very daunting today, but there's a lot of, I think, openings and avenues. And I think one of those is the realization of treating the right patients with the right drug. This is a lesson that was taught by a close mentor of mine that recently passed away, Phil Needleman, and thinking about rare diseases that we can employ these therapeutics to quickly understand whether they work and to see benefit. And so today we have a lot of cardiovascular diseases that we're really not good at treating. These include heart failure, patients that constantly get readmitted to the hospital that we really don't have options for, that particularly are not LVAD or transplant candidates. And then we have rare diseases, things like arrhythmogenic cardiomyopathy. They have a lot of inflammation and fibrosis that would be ideal patients receive therapies like this that really don't have any other options other than having an ICD and waiting for a transplant. And so we are very interested in using these different conditions in combinations with biomarkers and molecular imaging to identify individuals that have the biology we want to target going on in their heart. Individuals that have FAP positive cells, that have CCR2 positive cells in the myocardium, treating them with therapeutics, and then using the imaging to actually see if we hit our target. Do we remove the FAP positive cell? Do we suppress inflammation? We think that this paradigm will be a great approach moving forward. And so today we have lots of approaches for the FAP positive cells. We could inhibit IL-1 beta. We could think about these RUNX1 inhibitors or siRNAs. And I get a time to talk about it today, but there's also great approaches using bispecific T-cell engagers, bispecific macrophage engagers, and many other really cool antibody-based technologies to edit or eliminate the cells that exist within human tissues without employing cell therapy. So I think I've hit most of the take-home points. And so I'd like to end by just thanking all the people that make this work possible. This really takes a lot of individuals to do this. These are the individuals within my lab that directly contributed to the project: Junan Amrut, Stephen Yang, Alekia, Tyler, and Yun Ling, all graduate students, either MSTPs or PhD students. We have amazing collaborators, Rob Gropper, Yongshun Liu, and imaging. I didn't get to talk about our transplant work today, precision medicine, and then collaborators that really make translation and increasing the impact of your work a reality. And I'm really indebted to all of their efforts and generosity. And I'll end there and happy to take any questions. Thank you so much.
I'm mute. Sorry, I was mute. Thank you, Kory. It's a fantastic presentation, outstanding, really, as I expected, the state-of-the-art techniques to delineate particular molecular mechanisms in bigger picture of fibrosis. Before even we ask panelists to open the discussion, there are already questions from the members, including Christopher Mack and Sydney Chen. We will get to you shortly. Panelists, would any of you wish to discuss any particular topic? If not, we move on to the audience.
I have a quick question. What do you think is the origin of FAP positive cells in the heart? And what role does the RUNX1 pathway actually plays in FAP cells? Is it FAP dependent or is it independent in cardiac fibrosis?
Yeah, thank you. Great question. So we think the FAP positive cells come from resting fibroblasts that are quiescent within the heart, the so-called ground state, which matches them the best in forms of chronic heart failure or ischemic disease. However, there's other forms of heart failure we didn't look at, things like ICI myocarditis, which have a lot of endocardial involvement, where there could be other sources, and I think that remains an open question. The second question is when does RUNX1 function, right? Does it function in the quiescent fibroblasts, early activated fibroblasts, or once they're already activated. And so we're answering that now by knocking out at different stages using Cres that are activated either in the quiescent stage, early activation like periostin, or late activation states. And we're just learning the answers, but the clues that we're seeing at present is it may be acting in all of the different time points for its initial specification and maintaining that specification. And I think that speaks to how plastic these cells are.
Nick.
Kory, thank you very much for this great talk. I will start with a more translational question related to IL-1 targeting. So of the targets that you mentioned and you discussed, IL-1 seems to be the more translation ready. There is an experience with IL-1 blockade in models of heart disease for more than 20 years, always almost positive. Then there are, as you mentioned, great agents to inhibit the system. There is also from the Canto study, Paul Ritker had done the analysis of the effects on heart failure, and he had like prevention, had data in that circulation paper that appeared a few years ago that there were beneficial effects in preventing first heart failure events. So it seemed to me always like a natural target and a no-brainer for the industry, and we are dependent on industry on this, to pursue. Yet this not only has stalled, but the last thing I heard, and I never understood why, was that there was not particular interest on the part of key industry players who hold the agents of interest towards advancing this in heart failure, in post-MI setting, perhaps. Why is that? Do you have a better understanding than I do? Is it because they are always it's challenging to target heart disease, but that shouldn't be a deterrent by itself. Is it because they are afraid of stratifying patients, which they may need to do, and it's more expensive? Is it something else? What have you heard about this?
Yeah, these are great questions, and all the questions that we're asking ourselves too. So one, I agree that this is the right pathway that's translational ready, and it should be moved forward. I think there's a ton of biologic data, clinical trial data, genetic data that all speak towards the benefits of inhibiting IL-1 beta in heart failure. And there's many people to thank, including Paul Ritker, for making that happen. My understanding, it's largely a commercial issue that Novartis got very interested in cancer when they saw the initial data from Cantos that lung cancer was cut into like a quarter, and they ran three trials, Canopy 1, 2, and 3, which were all negative. And at the end of the day, they didn't feel it was worth they didn't feel like it was worth the money to develop in the heart failure space. I think there was also a feeling from industry, largely commercial industry, that heart failure patients would not be interested in taking injections, and there'd be poor uptake. So obviously Ozempic has proved that one wrong by a lot. And there's actually a resurgence now of companies interested in repurposing IL-1 beta inhibitors in indications like heart failure, knowing that with positive clinical trials, and we've been lucky to have positive clinical trials in the past 5 years in both HFpEF and HFrEF, and I think that's reignited the fire. I think we'll start seeing some of these studies come forward, and I'm not a big fan of IL-6 inhibition, but I think Novo Nordisk has set the stage for taking that risk, and I really hope other companies will follow suit.
Thank you very much for these insights. And the question now on the cell biology. So you show that this FAP positive fibroblasts are injurious. Why are they so much more injurious than other fibroblasts? Is it simply because of matrix production, of protease production? It's probably not FAP, right? I would think that it probably doesn't play much of a role. So there must be something else. And why are they also so responsive to IL-1? Did you find something to explain this, perhaps at the receptor level or something downstream?
Yeah, so we don't know all the answers yet to this. So that's a key question, right? Why are they have more of a negative impact than myofibroblasts or another population? I so a few things that they do. They make a lot of collagen, as much or more than myofibroblasts. They there are a few studies that have inhibited FAP enzymatic activity, and there's small benefits to that, but I don't think it's enough to explain the benefit we're seeing. They also make chemokines that could recruit other immune cells, monocytes, macrophages, and T cells that could amplify the inflammatory process. And I think that's probably the latter. We're currently doing a few proteomic experiments at present to try to understand at the protein level what their mediators are so that can be teased out genetically.
Thank you very much.
Thank you.
Michael Schubert.
Yeah, Kory, that was beautiful work. Thank you very much for that presentation. And I have two questions really on the cell population data. So Jeff Mole contains data on matrofibrocytes. How do those align with your 13 populations? Are they related to the FAP periostin population? Is it something else? Did you see that population within your set? Is my first question.
Yeah, so Jeff asked me that question all the time, as you would expect. The problem is, and it's not, I mean, it's just the technology that when he did it, right? He published that really in the pre-single cell technology time. And so the only markers he looked at were periostin and a few others. And so when we try to co-localize that set, the set of markers he kind of focused on, it does mark the FAP positive population, but it marks others too. It kind of nicely mirrors Taiwan. And so there's just not the resolution to look at it yet.
Right. So if you're looking at some of the markers like COMP or SILP or things like
Yeah.
that, it's kind of scattered. And you'd mentioned, I'm sorry, I'm just going to pull up my note here. So you'd looked at whether the FAP periostin cells could become the fibroblasts, and you said myofibroblasts, and you said no. But what about the other way? Do the myofibs ever become FAP positive, periostin positive cells?
Yeah, good question. We don't know. The only way to strictly know is lineage tracing, using a myofibroblast Cree.
Right.
And we're actually doing that with my PhD mentor. It's kind of fun, Dave Hornitz now. And so they're looking at that in their model, and we're hopeful to use single cell technologies to address that question. I would
Brilliant.
suspect the answer is no, but I'll tell you the real answer when we actually know.
Gotcha. Thanks very much.
Thank you.
Very good. Now, thank you, everyone. We move on to a couple of questions from audience. Christoph Mack. Chris says, wonderful presentation. A general question: how does targeting macrophage fibroblasts reduce infarct size and improve cardiac function? Is there crosstalk from macrophage fibroblasts to cardiac myocyte that can influence cardiac myocyte cell death pathways and/or cardiac myocyte function, such as EC coupling? And/or is the relief of fibrosis sufficient to improve systolic function, i.e. relief from mechanical restraints? Question mark.
Yeah, thank you for the question, Christoph. So I think the key question is, do fibroblasts do more than fibrosis? And I think the answer is absolutely yes. It kind of gets to Nick's question as well. We think they play key roles in recruiting monocytes and macrophages and maintaining an inflammatory sort of milieu within these immunofibroblast niches. And we think that that consequential activity of effector immune cells, particularly macrophages within those niches, reduces cardiomyocyte function through cytokine signaling. Some of those are hypotheses, some of those have data behind them, but that's the working model that we're using.
Then we have the next question is from Sydney Chen from UC Colorado, University of Colorado at Denver. She asked, to what extent do you view interleukin-1 beta as a dominant immune-derived signal orchestrating the transition of FAP-positive fibroblasts from reparative to pathogenic phenotype and how does it influence compared to other axes such as TGF-beta, PDGF, or type 1 interferon signaling in chronic cardiomyopathies?
Yeah, great question. Thank you for asking that. So obviously, IL-1 beta has a dominant effect, but we'd be a fool to think that's the only signal. Absolutely. Nothing ever functions in isolation. We're beginning to work with this, but one of the keys is having an in vitro model where we can manipulate these factors but be able to maintain fibroblast plasticity to become an FAP or periostin state. So a lot of that's still in the works, but what we know is that FAP and TGF-beta don't have a lot of synergy, but IL-1 beta and TGF-beta don't have a lot of synergy. But IL-1 beta does have synergy with some other cytokines that we're kind of currently sorting out. So work in progress and a great question. Thank you.
Very good. And final question from Dr. Lila Ruhi from UT Houston. Thank you. Thanks for your excellent talk. Do these interleukin-1 responsive fibroblasts go back to be more hemostatic state once interleukin-1 beta signaling is removed, or is there epigenetic locking that sustains the pathological phenotype? How stable is this fibroblast state once established?
Awesome question. So what we know today by tracing the FAP positive cells when you withdraw a pathologic stimulus is they don't go back to normal, but they shift and they toggle to a different area on the curve that looks like an intermediate state. And so there clearly is some epigenetic, I would say maybe memory or encoding that sustains a pathologic phenotype and really speaks to the fact that therapies to prevent them from maturing need to be chronic and can't be used in an acute manner. So great question. And I think that's a key question for the field.
Thank you. Dr. Agarwal, DK,
Yes.
is it time to say a few words about the IACS? Close the session.
On behalf of the IACS North American section and overall, and on behalf of Dr. Marian, sincerely appreciate your generosity to give this lecture. It's a brilliant lecture, very informative. Thank you so much. And those who are on the panel here as well as who are there, I will be happy if you all become the part of our IACS as a member. I have a quick question, Dr. Lavine. Is there a differential expression or differential composition of these type of fibroblasts, FFE positive or periostin positive in heart failure reserve ejection fraction versus HPAF? Do we have any idea about it?
We don't know much yet. One of the problems with HFpEF is there's not a lot of tissue available, right? And whatever tissue you can look at is small LV biopsies in Europe or RV biopsies from the US. We're really excited to use our imaging tracers, I think, to be able to answer that question in more detail. And so we are learning using those imaging tracers that in some forms of HFpEF, mostly hypertensive disease, interestingly also amyloid, that there's a lot of FAP positive cells, but it's not in everyone. So it seems to be in certain subtypes of HFpEF. And we're very curious whether that can be part of the whole phenotyping of that very heterogeneous disease.
Good. Another quick question, if I may ask, is that although Mike Schubert and Lila Rui also asked that question, but is there any way endogenously to de-differentiate the FFE positive fibroblast back to the beneficial type of fibroblast?
Yeah,
and
so
endogenous way.
Yeah, you can do that by giving a RUNX1 inhibitor as a delayed therapy or deleting RUNX1. That definitely works and shifts them back closer to a normal fibroblast state. And we're currently exploring whether IL-1 beta therapeutics have a similar potential.
Thank you. Thank you very much once again, Dr. Marian. That's wonderful. Thank you for letting me speak for a few seconds.
Thank you all very much. We are grateful for everyone that attended to this session, and our sincere gratitude for Kory giving us an outstanding state-of-the-art really lecture using the modern technology to get insight into pathogenesis of fibrosis. Our sincere gratitude to everyone. Thank you all. You all have a beautiful day, and see you soon in 2026. in February. Natalie will correct us all the time and for next webinar. You all have a fantastic day. Thank you, Kory. Gratitude.
Thank you.
Thanks, AJ. Thank you,
Thank
guys.
you.
Thank you, sir. Really appreciate it.
Yeah, excellent
Appreciate it.
talk.
Thanks, everyone. Have a great one.
Thank you, Natalie.
Thank you.